everyone. My name is Cong. It's spelled as C, but pronounced as TS sound. I'm an assistant professor at the University of Michigan at the Department of Computer Medicine and Bioinformatics. I'm going to talk about a tutorial for inference of copy number aberrations using bulk and single-cell sequencing data.
I don't think you need the microphone.
Oh.
Is the microphone red? Do you guys want me
Do I need a microphone or? Oh, this one. This is better. Okay, cool. For the focus is on copy number aberrations, CNAs, or also called copy number variations, CNVs. There are duplications or deletions of DNA segment in the genome. And so as a human being, we have two alleles for each chromosome, two copies for each chromosome. But for many diseases, like we see aberrations, we see sometimes like more copies of chromosomes or some part of the chromosomes. And particularly in cancer, there are a lot of CNAs that occur in cancer, and some of them are cancer drivers. And so here on this plot is basically showing a lot of important genes for cancer, oncogenes and tumor suppressor genes, and apparently a lot of the genes have copy number gains and losses, which is a very important explanation for how cancer occur and how cancer progress. And there have been studies to show that like copy number aberrations, like gains and losses, it can occur on all the chromosomes and occur on all cancer types. So it is like very prevalent and important in cancer studies. And more importantly, copy number aberrations also indicate intratumor heterogeneity. We heard of intratumor heterogeneity a lot, but what it means is basically like in tumor evolution, it's not like it doesn't directly grow a tumor at the current stage, but instead it gradually evolve and get more and more severe. And so it usually start with one tumor cell and then gain some genetic mutations, and those tumor cells expand, grow, proliferate, and then genetic mutation doesn't occur at a defined time, it can occur anytime. So new genetic mutation can also occur at a later time in some of the tumor cells, and so in the end we get a tumor population where each cell may have distinct sets of somatic mutations. And of course, like copy number aberrations is a very important type of genetic mutation we're looking at. And so usually like these genetic heterogeneity, it's referred to as tumor clones, where each clone is a population of tumor cell with an identical set of genetic mutations. So like heterogeneous tumor population means that we have a lot of different tumor clones or subclones. And here I don't distinguish the two words, clones or subclone. And it's important, tumors, it's important to know about intratumor heterogeneity. Tumor likes to have like a lot of different genetic diversity. It's benefit tumor, bad for patients. So here there are like previous studies showing that if you have multiple different clones in the tumor, it's very likely that like once got treated, it will relapse. It has like a lower rate of being free of tumor, so higher rate of tumor getting back. And also here, lower rate of not having metastasis, so like having more clones means where it's more likely to have metastasis to other tissues and organs. And so it's important to use copy number aberrations to study potential why tumor occur and also the intratumor heterogeneity. And so the question is, how can we infer copy number aberrations from data? And so there are a lot of sequencing data, sequencing technologies has been developed, sequencing data has been published for, I guess, tens of thousands of cancer patients. There are a lot of different types, DNA sequencing, RNA sequencing, and methylation, etc. And the question is, given the sequencing data, can we infer this copy number aberration profile of the genome of the tumor? And before going into how we can do that, let me clarify two more concepts. Total copy number aberration, total copy number, and allele-specific copy number. And so we have, as I said, we have a parental allele a maternal allele and a paternal allele, like two copies, each from one of our parents. And so total copy number means just like directly sum up the two copies from two parents. And allele-specific copy number means we're distinguishing between the two parental allele and count the copy numbers of each parental allele separately. And so it's important to distinguish between the alleles because it reveals more of a comprehensive picture of what the copy number aberration look like. And so here on the toy example, can I use this? Oh yeah. So in the toy example, if we only look at total copy number, here a toy example of normal cell with two copies in this chromosome, and we may also have a tumor cell with two copies in this chromosome. It doesn't mean that this chromosome doesn't have copy number aberrations in tumor, but instead, if we look at allele-specific copy number, we may see that in the normal, we do see one maternal copy and one paternal copy, but in the tumor, we may see loss of maternal copy and two copies from father side. So this is known as copy number neutral loss of heterozygosity. It's like we see that a lot in TCG cancer studies. It's important to be able to capture this if we want a full picture of what copy number aberrations are there in each patient. And it's also here like two tumor cells with three total copy numbers. We may think that they share the same ancestor that have a copy number gain in this chromosome. But instead, if we look at allele-specific copy number, we may see a copy number gain at different parental alleles, which means that the two tumor cells actually undergo two independent copy number gain events. And so it's important to not only infer the total copy number changes, but also to infer the allele-specific copy number changes. And so that's also a lot of previous work has been done to infer allele-specific copy number aberrations. And more about the data we're using. I think we heard about like whole genome or DNA RNA sequencing a lot, but I just have the slides so that we're on the same page. That like all the sequencing technology allow us to read out the ACTGs on our genome or on each of the sequencing reads. And most of previous data, they use short read sequencing. It's about 50 to 300 base pairs. So it's like compared to our genome 3 billion base pairs, it's a very short region. And so we need to like sample a lot, like the tens of thousands or even millions of short reads, so that it will get like a high coverage of everywhere in our genome. And there are a couple of different differences in different sequencing technologies. For example, whole genome sequencing, it will have relatively uniform coverage across all places in our genome. Whole exome sequencing, it only covers the exome region, but it will still have relatively uniform coverage. But compared to RNA sequencing, it of course only covers the gene region or most likely exon region, but it will have a very drastic coverage difference across different genes because gene expression are very different across different genes in different cell types. And so with this sequencing technologies, how we can tell which part of our genome have potential copy number gains and losses. As I said, like tumors can have intratumor heterogeneity. But let's start with something simple that like, let's assume that like we're dealing with a sample that have homogeneous tumor cell population. And let's suppose like we have a ground truth of what the copy numbers are, allele-specific copy numbers are in this genome across all chromosomes. And so suppose we're doing a sequencing using this on this group of cells. For example, whole genome sequencing, what we may see is if this region has a higher copy number, then it's more likely to sample a sequencing read from that region. So in the end, if we align the read back to the genome, we may see more reads aligned to this region with higher copy number compared to those with normal copy numbers, or even higher compared to those with the deletion in the DNA. And so right now, if we're getting this alignment of the sequencing reads back to the genome, and we see relatively higher coverage of like a higher read depth of this region compared to other regions that indicate potentially this region has a copy number gain. And so a lot of past literature, people developed the metric read depth ratio to indicate the potential total copy number, the difference between tumor and a normal reference. So it defined as the ratio between the proportion of reads mapped to a specific genome loci or in a genome interval compared to the proportion of reads mapped to the same region in the normal sample. And so we use the word proportion because we're trying to avoid the complication from different number of total sequencing reads, or what's it called, library sizes. And if we have a large number of sequencing reads, the proportion of the reads will converge to the proportion of DNA base pairs. And so that, or DNA base pairs, it's also indicated by the copy number of that DNA region. And so we can relate this read depth ratio, RDR, with the copy number in the DNA that we are trying to infer. And so here, it's a ratio between proportion of reads in the tumors, or it will approximate the proportion of DNA copies in the tumor, which is the sum of copy number at this interval I compared to all copy number. It's like the copy number of like all regions. And in the denominator, we have the proportion of copy numbers at this region in normal sample, where we assume in normal sample it's nothing changed, so all copy numbers are 2. And so we further rearrange this equation. We have RDR, it's approximately equal to the A allele copy plus B allele copy, or the total copy number at this interval, divided by the average copy number across the entire genome, which is also known as ploidy. You may see in some literature, instead of using like raw RDR, they use log RDR or log R, which is like log transformed of the the ratio of read proportions, and right hand side it will be the log transformed version of this ratio. And as said, this is RDRs for total copy number gain and losses. As said, we want allele-specific copy number. So how to distinguish between alleles? So we use, or people have used heterozygous SNP loci to distinguish between the two parental alleles. And so heterozygous SNP loci, that means mother allele and father allele, they're different. They're heterozygous by nature, like heterozygous when they're born, and without copy number aberrations. So if we do a sequencing on the a normal cell, we would expect 50% reads will come from one of the allele and 50% reads coming from the other allele. But if there is copy number aberrations, for example here, the orange allele, the B allele got deleted, and so that all the sequencing reads will only cover nucleotide G without seeing nucleotide T. And so using heterozygous SNP loci, we can approximate the relative ratio between the copy number of the two parental alleles. And so we use B allele frequency, or BAF, which is defined as the proportion of sequencing reads from B allele at germline heterozygous loci compared to the sum of sequencing reads covering covering both parental alleles. And we can also relate the B allele frequency defined using the sequencing rates with an expected value from the copy numbers, which is it will approximately equal to the B copy number over the sum of copy number at each SNP loci I. I guess if the number of reads is large enough, the approximation would be good. So this is given a specific copy number profile, we can derive what we expect the RDR and BAF value to be. But here we don't know the true copy number in a general new sample. But instead we have the sequencing data where we can compute RDR and BAF of each genome loci, and we can take these as input to back infer the allele-specific copy number, the A copy and the B copy at each genome loci. And so in this way, we know RDR, how RDR is related to the copy number and how BAF is related to this copy number. And we can also impose some distribution assumption to this relationship, and then inferring A and B copy number becomes a parameter estimation problem under this probabilistic model. We can do this, technically we can do this separately for each genomic loci, but additionally we have a prior belief that copy number aberrations, there are copy number gain losses of a region, and usually a large region. So we would assume copy number are coherent along the genome, and it's very likely that between two adjacent genome intervals or genome loci, the allele-specific copy numbers are equal to each other. And so instead of estimating A copy number, B copy number for each genome loci separately, we can first partition the genome into several chunks such that for each chunk we believe that they have the same copy number, and then estimate a allele-specific copy number for each chunk separately. And this can be done by many change point detection or segmentation algorithms. There are a lot of work on this algorithm. Another line of idea is we can do the segmentation, but instead of using previous change point detection method, we can do a clustering, a locality-aware clustering using hidden Markov model, where hidden Markov model will have a hidden state, and in our case, the hidden state represent the allele-specific copy numbers, or it's a cluster of the genome loci for which the genome loci will have identical copy numbers in each cluster. And so given this cluster label, all the observed RDR and BAF will share the same parameter, allele-specific copy number in each cluster, and instead we're inferring the clustering label and optimizing the RDR and BAF, observed RDR and BAF together. And also we can also impose the cluster label or hidden state to be coherent in the genome. And so here is an example of cluster or hidden states inferred by HMM on a real data. and here each color indicates the cluster label. And in this case, we not only have a segmentation of the genome, but also can relate distant intervals, the segment in the genome, and say that they actually have the same copy number, belong to the same cluster. So this is, I guess, on the ideal case. Suppose our sample is relatively homogeneous. We can infer the copy number using combining the segmentation and also the parameter inference of the probabilistic model. But in general, we're dealing with more complicated data where each sample is it's a mixture, potentially a mixture of normal cell and tumor cell, and potentially also a mixture of tumor cell with different subclones. And in that scenario, how we're going to infer copy numbers. Our signal from the sequencing reads, RGR and BAF, it's still defined in the same way. It's not affected because it's still like from the sequencing reads. But then it's still they're still in proportion to the copy numbers. But then the copy number will not be a copy number from a pure sample, but instead the average copy number across the mixture of population. And so here, the RDR, the numerator of RDR should be a weighted average of the tumor copy number and also the normal cell copy number, which is 2, and also same for biliary frequency BAF. And in general, if we have multiple tumor cell populations and we have the mixing proportion of the tumor cell in each clone, and then we can further express RGR and BAF quantity, relate RGR and BAF to the weighted average of the allele-specific copy number within each subclone. So this is actually the core quantity we can use to infer copy number across many different data sets, data types. For example, bulk DNA sequencing, one bulk DNA sequencing, we can also join inference of copy number across multiple bulk DNA sequencing sample, single cell RNA sequencing sample, and spatial transcriptomics sample, and so on. But each of them may have their own considerations for the different constraints on this core assumption. For example, in one bulk DNA sequencing sample, it will be hard to infer multiple different clones from the same sample. So typically, we can only reliably infer at most 2 copy number from 2 tumor clones. And if we have multiple bulk DNA sequencing sample from the same patient, it's the same patient, so they should have the same tumor clones, but potentially with different proportions. So that means copy numbers are shared across samples, but the clone proportion are different to each of the sample. Also, single cell RNA sequencing sample, I haven't talked about this data, but based on its name, it's single cell, so it doesn't have the tumor population mixing problem. And so each clone proportion, it will be binary, it will be either 0 or 1. And for spatial transcriptomics, if the spot is small enough, we can also assume the tumor proportion to be close to binary. In the interest of time, I think I'm going to skip the details for inferring copy number from bulk DNA sequencing data, but directly jump to spatial transcriptomics because that's newer. So spatial transcriptomics, there are many technologies. Sequencing reads RNA molecules from thousands of spatial spots and also retain its 2D spatial coordinates. And essentially in the end, we can get a gene expression matrix of spot by gene expression matrix. But in fact, we also have, for some technologies, it's based on actual sequencing, so we can have the raw sequencing reads and can still get the SNP information and get the alleles. And but spatial transcriptomics data has a challenge that the rate count is really sparse. And I said that the B allele frequency is a proportion of reads from B allele divided by the number of reads at both alleles. But here in spatial transcriptomics data, it's extremely sparse, and at each, for each spot, at each genome loci, the total number of sequencing reads covering the SNP we're looking at is about the integer scale. Most of them are actually 0. So it's not possible to have like a really like proportion when the denominator is 0. So this is very problematic, it's an additional challenge in spatial transcriptomics data. But luckily, if we can group all the spots into, group spots into pseudo-BOC, and compute the B allele frequency across, I guess, a mixture of many spots, then we're still able to see some signals. Here, the deviation from 0.5 indicates potentially some allele-specific copy number gain and losses. And so we need to do something special to handle the sparsity of the spatial transcriptomics data, and specifically all the ratios. Like we cannot actually using ratios anymore, and instead we resort to count-based probabilistic distributions to directly model the counts rather than the ratios. And so here, the transcript counts, which will correspond to the numerator part of the RDR, we may assume that it follows a negative binomial distribution where its parameter or its mean parameter is a multiplication with the expected count if the spot were normal, and also scaled by the RDR parameter or the theoretical parameter from the copy numbers. And also similarly for B allele frequency, we'll directly model the B allele count using the total allele count, assuming it follows binomial or beta binomial distribution, where the parameter P, the BAF parameter, will correspond to the theoretical BAF value derived from the copy numbers. And so additionally, typically like copy numbers are rare. We don't have a lot of copy number, or a different number of tumor clones can be distinguished by copy numbers. And so usually, here the clones, we don't have a large number. We only have a handful of tumor clones maybe. And so for the spot, like which spot has clone C, or which spot has clone maybe of another clone, this is unknown. And so we would also need to infer that if we want to infer copy number in spatial transcriptomics. And so we need to further incorporate a new variable, called clone label that indicate for the clone proportion which clone has a proportion of 1, which clone has a proportion of 0 at each spot. And so we assume, it's spatial data, we assume the clones expand locally so that adjacent spots have similar clone labels. And also, if we had that, all like copy numbers, hidden marker model assumption follows the previous work. And so we're eventually combining these assumptions we can combine this into a joint probabilistic model to combine the hidden Markov model to model the copy number along the genome, and also the tumor clones in space, so that we can also enforce prior distribution assumptions so that copy numbers, they are coherent in the genome and the clones are coherent in space. And so finally, this is a cool visualization of real data results that like we have a prostate cancer, 5 spatial slices, and we can, based on that framework, we can infer allele-specific copy numbers and tumor clones together across these 5 slices. And so this is, we're able to identify 5 clones, and each of them have their own allele-specific copy number profiles. And finally, here is the clone in space. We can see like some of the clones are usually spatially coherent and they occupy different spaces. And using the mutations, we can also reconstruct the phylogeny, which I think you will hear more about it tomorrow. So the phylogeny here indicates the tumor may start somewhere in the middle of the prostate organ and move to the left and right side during tumor evolution, and finally move to the very left edge and very right edge and become very severe stages at the time of sequencing. And so here is the summary. I introduced read depth ratio and allele frequency to indicate allele-specific copy numbers. I talked about spatial transcriptomics, and we can infer allele-specific copy number from spatial transcriptomics using a coupled hidden Markov field and hidden Markov model, and some references. Okay, thank you.